Podcast number 17, and in number 17 we're going to talk about more evidence for how abiotic vesicles, so no biological component, they're not parts of cells or anything like that, but they have macromolecules, how they can sort of act like cells. And if we can model this in a laboratory, we might get some idea how the first cells came to be on Earth. So one of the molecules that we talked about in the last podcast is a phospholipid. It's called so-called amphipathic, AMPHI, pathic, in that it means it has a part that does not like water. This tail here does not like water, and a part that does like water is hydrophilic, this head group here. And when you put a bunch of these in water solution, they can form a single layer of all the phosphate heads are pointed out and the tails are pointed in. That's called a micelle, or a double layer where you have two layers of phospholipid forming this vesicle. And when you have a vesicle, this is essentially the building blocks of a cell membrane, where you have the outside components of the cell here. This would be like blood, and then the inside components of a cell. This is the cytoplasm, where this would be like cytoplasm, where the parts of the cell are. So can we get these things to behave almost like cells? Well, one of the things that we associate with cells is growth. Can you get them to grow? Some early experiments found that clay and charged ceramic material and clay that had a negative charge on it could act like a catalyst, in that it could cause the formation of growing of these vesicles faster than if there's no clay there. So here's an experiment where they measure these things scatter light in solution. They are essentially fat, and fat mixed into solution makes it cloudy, and so that will absorb light, and the more light absorbed means higher concentration of these things in solution. So over time, if you don't add ceramic microspheres, these vesicles do increase over time, but they increase lots faster in the presence of ceramic microspheres or clay particles that have a charge on them. Not if they don't have a charge on them, but if they do have a charge on them. The shape of this kind of curve and the shape of these kinds of curves here look very much like how enzymes work and how fast the enzyme works over time. And enzymes are products of translation, they're products of cells, whereas these are just chemicals put together. But the scientists are trying to show, look, it looks just like an enzyme. In this case here, the enzyme increases dramatically, and then it starts to tail off a little bit. And this is also a property of an enzyme. If you can think of an enzyme, I like to think about them, a visual in my head, as like those competitive hot dog eaters. So in the case of the competitive hot dog eater, the enzyme, their job is to chew up the hot dog, and the substrate that they work on is hot dogs, and the product is whatever hot dogs make in a belly. We won't even talk about that. If you think about a competitive hot dog eater, they have to have a lot of hot dogs in order to keep going. And if you take the hot dogs away, they slow down because there's nothing to eat. Same thing with enzymes. Enzymes start off going really, really fast as long as there's plenty of substrate. But as they run out of substrate, as they run out of hot dogs to eat, they slow down in making a product, the hot dogs in the belly. So these aren't enzymes. These are just vesicles and clay, but they're sort of acting like enzymes. So if we look at this close again, here's our amphipathic molecule, has a hydrophilic water-loving part and a hydrophobic water-hating part. They can form one layer or two layers, like in a vesicle. And when added to ceramic microspheres, they get bigger. But if the microspheres have a negative charge on them, they get much, much bigger, faster. And the shape of these curves looks like the shape of an enzyme curve in terms of speed over time. This isn't speed, they're measuring just the formation of these vesicles, but it looks just like an enzyme curve. Okay. And if you don't add any clay, they get bigger over time, but not a whole lot bigger over time. But if you add more and more vesicles, okay, they go faster and faster. And you can't compare the whole curve because at this point, your hot dog eater is not at full capacity. They're not going as fast as they can. So normally, when you're measuring an enzyme, and again, these aren't enzymes, but they're acting like enzymes, you measure it just as the reaction is just starting. So in this case, in the first part of all these curves, they haven't run out of hot dogs, or they haven't run out of vesicles, raw material, that phospholipid to put together into vesicles. They haven't run out of that. And so they're going as fast as they can. And so you want to compare the conditions where things are going as fast as they can. And essentially, the more substrate you add, this would be phospholipids, or in the case of our analogy, hot dogs, the faster it goes. Okay? These have the highest concentration of phospholipids and they go the fastest. And if you just look at the slope, okay, so rise over run, how fast are they increasing versus time, that's a ratio, and if you just look at those ratios, it's directly proportional to the concentration of the clay that they add. So as long as there is enough hot dogs to eat, the concentration of clay acting like a catalyst makes the reaction go faster. And the point of these things is these things act like enzymes, and all of this stuff can be made abiotically. Like we talked about in the last podcast, in the Miller-Urey experiment, where he just boiled stuff in his apparatus, he got things like phospholipids. NASA has also done experiments where they can show that UV light shining on surfaces make these phospholipids. And these phospholipids, this thing here, I didn't tell you that's important, is they tend to collect on particles of clay. So you can get a big enough concentration to create one of these things or one of these things. If they're just floating around in the big old ocean, there might not be enough of these made, but they tend to collect on clay, sort of like if you drop a bit of your corn chip that you're eating into your soda, the bubbles will collect on that corn chip floating on your soda, and these things will collect on clay particles. So these things could be made in the early atmosphere of the Earth and on the surface of the Earth, and clay was around in the early surface of the Earth, and it's thought that clay acted like a catalyst to make more and bigger of these things that look like cells, at least have the right shape of cells and the components of cells. Okay, but now you've got this microsphere. Can they do anything else? Well, they dyed them green and then they fed them different things. Like if they feed them clay particles, those clay particles show up inside the beads. If they feed them pieces of RNA, those RNA pieces show up inside the beads, or inside the vesicles. And so now you have something that even looks more like a cell in that it has a membrane surrounding it of phospholipids and then RNA on the inside. And then we know from the last podcast that RNA can act like a catalyst, like an enzyme, and it can further catalyze other biochemical reactions. Another thing we associate with life is growth. So can these pseudo cells, these vesicles that we're saying may have been some of the first cells, can they grow? Well, if you look at the size of the vesicles when you first put them in solution, and then after you feed them more phospholipids, they get bigger if you feed them more phospholipids. And feed them, it just means you add more to the solution, causes the vesicles to get bigger. Okay. And if you give them more time, they get bigger and bigger over time. Okay, and that's another thing that we would think about as how cells growing. Can they divide? So we just talked about last week mitosis and meiosis. Can these cells divide? This you got to use your imagination for a little bit in terms of, are these conditions similar enough to a cell in early Earth? So if we start with a bunch of vesicles and they're about this big, when you start the experiment, then you give them more micelles, more phospholipids, and they get bigger. Then you force them through a filter, and they get smaller, and they divide when they get smaller. So this is one big one, and this would be two smaller ones as a result of that. It's not like this big one just got smaller. It actually divides in two. And then you can go through several cycles of this abiotically. So this division here that is happening in this experiment is by forcing these vesicles through a filter. Now that's not what happens during mitosis and meiosis. For sure, that's not what happens. You saw that it's a pretty complex process, involves lots of different parts of the cell coordinating together to divide the cell. They are saying, these scientists are saying these are similar conditions to what these vesicles would go through in like a shallow puddle. That as the puddle dried out and then filled up with exposure to heat and then rain, they would go through a similar process of what they used in the lab to divide these cells. So it's a demonstration that it's possible that some abiotic force on the Earth could have made these vesicles. These vesicles could accumulate things inside of them, like the RNA. The RNA can act like an enzyme, and then these vesicles could divide, could grow, and then could divide, all with abiotic forces. That's a possibility. Okay, another aspect of life is storage of energy. One form of energy storage is in the storage of a pH gradient. And so we need to talk a bit about pH first. Sorry, this arrow is out of place. It's supposed to be over here a little bit. So you've heard of the term of pH. Your shampoo is pH balanced, or your maybe your soap is pH balanced. It's a way of measuring acidity or alkalinity of a solution. And what that is in definition is that water molecules will split into H plus ions and OH minus ions in solution. And if you measure the concentration of the H plus ions in a sample of pure water, it would be 1 times 10 to the minus 7 moles per liter. So moles, you might remember from chemistry, moles is a unit of measure, like a bushel or a gram or something like that. It's 6.02 times 10 to the 23rd particles, and in a concentration of pure water, there are 1 times 10 to the minus 7 moles per liter of H plus ions and the same number of OH minus ions. At lower pHs, the H plus ion concentration goes up higher than 1 times 10 to the minus 7. And that causes the pH to go down below 7. And at higher pHs, the OH minus solution goes up, concentration goes up, and those are higher pHs. So people tend to talk about pH in terms of acid, but there are all kinds of things that you encounter in your life where pH matters. So here's a pH scale, and it's a log base 10 scale. And how you interpret that is log base 10 is 10 to the minus 7 molar concentration of hydrogen ions. That is a pH of 7. Okay, and a pH of 2, for example, would be 1 times 10 to the minus 2 concentration of hydrogen ions, and 1 times 10 to the minus 7 is much less concentrated than 1 times 10 to the minus 2. And so if you look at pure water, this is versus lemon juice. Lemon juice is much more acidic, has a much higher concentration of hydrogen ion concentrations. Okay, and because this is a log scale, every time you go from like 7 to 6, 6 to 5, 5 to 4, you're increasing the concentration of hydrogen ions tenfold. So pH 6 versus pH 7, 6 has 10 times more hydrogen ions than 7, and 5 has 10 times 10 times more than 7. So this is 100 times more concentrated in terms of hydrogen ions versus 7, for example. Okay. In the other direction, hydrogen ions go down in concentration, but OH minus ions go up. And in either direction, they can be bad for biological systems. So gastric acid outside of your stomach can cause burns. Bleach outside at higher concentrations can also cause burns to biological systems. So everybody thinks acids are the bad things. Strong acids or strong bases on either end of the scale is a bad thing. Okay, so now you know that it's a log scale. Every time you go one number on the log scale, you're changing the hydrogen ion concentration tenfold. From 7 on down, increasing hydrogen ion concentration. For 7 on up, decreasing hydrogen ion concentration. Okay, what does that have to do with if cells and these model vesicles can store energy? Okay, phospholipids have a charged outer head and hydrophobic tail, and then they set themselves up so the tails are facing each other and the heads are facing water. In a water solution, there can be free hydrogen ions because water breaks into H plus and OH minus, and those free hydrogen ions can interact with these head groups. In a solution where you have a micelle, like this, or a vesicle like this, you can add new phospholipids to that. Those new phospholipids may carry a charge with them, and then they can flip from one side to the other and dump off their charge, their H plus charge on the inside. So by this mechanism, okay, originally the charges are balanced, new lipids fuse with the outer layer, some lipids might carry a hydrogen ion, those flip to the other side, and they dump off their hydrogen ions on the inside. By that mechanism, hydrogen ions move from the outside of the cell to the inside of the cell, and hydrogen ions, and it's not a cell, it's a vesicle, sorry. Those hydrogen ions can build up on the inside of the vesicle, and then you can end up with a pH gradient where the pH on the inside is lower than the pH on the outside, and that's a gradient of concentration gradient of hydrogen ions, and cells use concentration gradients of hydrogen ions to generate energy. We're going to talk more about that later, but that's a common mechanism that cells use to generate energy. So these are not cells, and if you take vesicles and then you put them in the situation where you add phospholipids to them, the pH on the inside of those vesicles goes down over time. So they're accumulating hydrogen ions, causing the pH to go down on the inside. Okay, first quick pH reduction after adding micelles to the vesicles, and then a slow, long-lasting pH reduction. Okay, if we look at this whole thing, and again, the theme here is, can you make something that looks like a cell from abiotic forces, where you're not making a cell from all of the cells, you're saying where did the first cell come from? And is it possible to make something that sort of acts like, looks like, walks and talks like a cell in a lab, starting with inorganic materials? Well, you can make nucleotides and lipids abiotically, as shown by NASA experiments and also the Miller-Urey experiment, that the abiotic RNA genome can replicate. RNA can act like an enzyme and catalyze itself to make more copies of RNA, that the RNA can catalyze vesicle formation. I didn't show you those data, but if you add RNA that gets sequestered inside these vesicles, it actually helps making more new vesicles. They can compete and grow for resources, more phospholipids, and they can grow. I didn't show you the competition experiments, but they can grow. If you force them through the filter, they can divide and reproduce. I didn't show you that evidence for volume grows faster than the surface area. The RNA that is in the environment can be taken up by vesicles, as shown by these experiments that labeled phospholipids in green and RNA in clay in red. The RNA gets taken up by the vesicles. Because vesicles have RNA and RNA can catalyze vesicle formation, they have an advantage of getting new phospholipids to make them bigger than ones that don't have RNA. The RNAs that replicate faster, you can do, we showed you how artificial selection can generate RNAs that replicate faster, so we know that it's possible that selection can produce RNAs that replicate faster. And then in this last couple slides, it showed you that abiotic vesicles can store energy in the form of a pH gradient by pumping hydrogen ions into the middle of that vesicle. Now the concentration of hydrogen ions inside is higher than outside. That creates a gradient across that membrane. And you'll see soon that gradient is used by cells, modern day cells, to generate energy.